Hi all. In this video, I am going to discuss about a phycology topic that is Spirogyra, its structure and reproduction. It is a freshwater form and the filaments are found as free floating. Filaments are simple and unbranched, cells are longer than broad. All the cells are alike and there is no distinction into base or apex. The cells have cellulose cell wall and covered with mucilage sheath which makes the filament slimy to touch. The protoplast of each cell contains cytoplasm, a single nucleus and one or more chloroplasts. Streaming movements are seen in some species. The nucleus suspended at the center by cytoplasmic strands. The chloroplasts are ribbon-shaped and spirally twisted. They show many pyrenoids embedded in them. The chloroplasts are narrow with a smooth margin or broad with serrated margins with scattered pyrenoids. The cell sap is rich in tannins. All cells of the filament are capable of cell division followed by elongation. In this slide, first diagram is filaments of Spirogyra in low power. Second one is detailed structure of Spirogyra. Next we are going to see about reproduction in Spirogyra. There are two different types of reproduction in Spirogyra, that is vegetative and sexual. Vegetative reproduction. While floating, long filaments broken into small fragments. Each one grows into a separate filament. This is known as fragmentation. Here we will see sexual reproduction in Spirogyra. Sexual reproduction by conjugation. The conjugation taking place between two cells of opposite filaments is known as scalariform conjugation. The conjugation takes place between two adjacent cells of the same filament known as lateral conjugation. Scalariform conjugation is very common in Spirogyra. Scalariform conjugation in detail. In scalariform conjugation, the filaments lie parallel to each other in a common mucilage. At the point of contact, opposite cells produce peg-like outgrowths. They grow into tube-like structures and push apart the opposite filaments. The end walls at the point of contact dissolve, forming a passage between the opposite cells, and a conjugation tube is formed. The entire protoplast of each cell shrinks and get rounded up into a single motionless gamete called aplanogamete. One of the gamete moves from its cell into the opposite one by an amoeboid movement. The migrating gamete is the minus type or the male one, and the other stationary gamete is the positive type or the female one. The two gametes are structurally similar but functionally dissimilar. The male gamete fuses with female gamete forming a zygote. Lateral conjugation. A lateral protrusion is produced by a cell of a filament at the septum. The septum dissolves and a passage is formed between adjacent cells. Male gamete moves into the female cell through the conjugation tube and fuses with its gamete forming the zygote. This diagram shows the scalariform conjugation in Spirogyra. In this picture, we can observe lateral conjugation in detail. Zygote. Cytoplasm of both gametes fuses and followed by delayed nuclear fusion. Zygote stores food and develops a thick wall to overcome the unfavorable conditions. Germination of the zygote. Zygote released by the decay of the cell walls. It germinates when conditions are favorable. The diploid nucleus divide meiotically first, forming 4 haploid nuclei, and one remains and others degenerate. The outer thicker wall of the zygote bursts and the inner wall grows into a long tube which undergoes transverse division into 2 cells. The lower cell contains scanty chlorophyll and it develops into a rhizoid. The upper cell continues to divide and a filament is formed. In some cases, conjugation may not lead to fusion of gametes. In this case, the gamete rounds off and secretes a membrane resulting in azygospore. Example, Spirogyra mirabilis.